For this final assignment, I'm going to be talking about the structural activation mechanisms and social determinants of health, how they go hand in hand. So, a little introduction to MAP kinases. MAP just stands for mitogen activated proteins. These kinases are activated by phosphorylation, as you can see in the left diagram over here. There's a whole cascade. So there's an external stimulus which stimulates MAPKKK, which is mitogen activated protein kinase. And then that stimulates MAPKK, and that phosphorylates MAPK, which can also phosphorylate downstream substrates such as other kinases, or it could also act as a or it can phosphorylate transcription factors, which affect gene regulation, gene expression, or gene regulation, and these can also have cellular effects, cellular consequences, or um, responses, such as airway inflammation and remodeling, which are characteristic of COPD and asthma. So MAP kinases are major components for not just asthma but all kinds of processes, such as embryogenesis, cell differentiation, proliferation, growth, and death. So these are very widely seen across all kinds of cancer pathways. That was one of the major ones. And to the right we have a diagram of, I think it's an X-ray crystallography structure, of P38, which is our protein of interest, specifically the gamma form, isoform, I think. Okay, so now we're going to talk a little bit about MAP kinase activation. So MAP kinase is activated by phosphorylation, as I said earlier. Um, its active site here is this is ANP. ANP is I think it's a nitrogen added, instead of okay, I'm not sure about the structure of ANP, but in essence, it's an inhibitor of ATP, and it is used to study ATP function. So it binds here, but generally we would see ATP binding here. And how does ATP bind here? It In its inactive form, it's unable to do so, which is the diagram to the right. This loop encircled in red is called the phosphorylation loop or the activation loop. And on activation, it's phosphorylated. Two residues are phosphorylated. We have the tyrosine at 185 and threonine at 183. And these are negatively charged groups, the phosphate groups. So when they phosphorylate these areas, the surrounding residues are actually positively charged at physiological pH. That's because so I've described it in the next slide. We have arginine 176, arginine 202, and arginine 142, which are proximal to that phosphorylation lip. And these at physiological pH have a positive charge, so they're likely to attract the um, the the phosphorylated groups that I discussed. And strictly visually speaking, you can see that when they're phosphorylated, it's a lot more condensed and sort of attracted to the upper portion of this protein. Whereas where it's dephosphorylated, you see it's like a little more dispersed, spread out, and sort of blocks that active site that I was talking about. So yeah, what happens is when it phosphorylates, it moves by the activation loop. It moves by about 5 to 10 degrees, which is a major conformational change, and this creates a pocket for the binding of ATP, allowing for the binding of substrate and then further catalysis. So structure can be analyzed um, through various methods. I outlined a few of the basic steps that we could take. So functional assays could be first employed to to distinguish between the inactivated and activated protein. So how I envision this is we would we would use substrates downstream or upstream, potentially downstream actually, of the enzyme, and then we would use both phosphorylated and dephosphorylated. It would be a mixture because we're distinguishing it, um, and the phosphorylated protein would act on the substrate, whereas the dephosphorylated protein that is inactive wouldn't be able to perform its catalytic function. And uh, to a similar degree, the physiological level assays would be uh, would have would be performed a similar way, but they would have a physiological consequence rather than a cellular consequence. And I explained this a little bit in the next slide about what specific physiological consequences or effects there are. Um, but that's the basic rundown. Next are some structural assays such as X-ray crystallography. It's one of the more it's one of the more sophisticated structural analysis techniques. Um, although EM, so that's a little it's catching up, but the resolution is a little lower. X-ray crystallography is higher, so it could um, maybe capture that nuance that the phosphate group carries. It's a small group to be captured by imaging, so we would want a high resolution technique. And then analytical tools, of course, like Chimera X, could be used to explore structural features of the proteins. So this matters socially and clinically because there's a lot of minority groups like black people, indigenous communities, uh, low income communities that live in discriminatory zoning areas, which have been established by the U.S. So, and these zoning areas have been placed near industrial areas where there's a higher incidence uh, of 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 breathing hazards that can cause asthma or which can stimulate asthma or COPD. So yes, and environmental pollutants can overly stimulate the enzyme P38, as was described by Pillaya in 2020. And understanding structure enables us to design inhibitors for P38. And we're looking at specifically the gamma isomer or isoform. And we can modulate hyperactive inflammatory pathways. So this diagram is a summary of what I've been talking about so far about physiological effects and inflammatory molecules. So, as I described before, there's an external stimulus acting on MAPKK, which phosphorylates MAPKK, phosphorylates MAPK, and then that goes down to downstream substrates. This can have physiological effects like growth factors being secreted, inflammatory cytokines, and UV osmotic or oxidative stresses. P38 is inhibited by all these molecules, so BIRB 796, PH797804, and there's so many that can inhibit all three or four isomers of P38, alpha, beta, gamma, and delta. And these have downstream targets like MAP, CAPK. I know that one is a COPD or asthma. It influences that pathway, right? And yeah, it's a very complex pathway, all intercalated. Growth factors can also be have been can be affected by these three, but this TPL2 affects MKK47, so it's pretty complicated. Yeah, that's all I have. Thank you for listening to my presentation. This was question number 4. Thank you.